Hey guys! So this is going to be the video over Tricky Topics Chapter 10. And because Chapter 10 is pretty short and this is more of a building block for the other chapters, there's not a lot of information here. So this is only going to be one video going over the DNA and RNA basics. So let's get started. So first we're going to talk about the structure of a nucleotide. A nucleotide is going to be the building block in both DNA and RNA, and it has three parts. The first part is the phosphate, the second part is the sugar, and the third part is the nitrogenous base. You can see all of these right here: phosphate, pentose sugar, meaning there's 5 carbons, and nitrogenous base. And there's another term that's just a good one to know. It's called a nucleoside, and this only consists of the sugar and the nitrogenous base. So it'd be these two right here, no phosphate. So let's talk about that phosphate really quick. Phosphate is going to be the exact same in DNA and RNA, and it's going to be negatively charged. And this means that DNA and RNA are both going to be negatively charged. And for the sugar and the nitrogenous base, these are going to look different in DNA and RNA, so we're going to talk about those separately. So let's start with the sugar. So if we were to look at this little picture on the side, you'll notice that both are pentose sugars, those 5 carbons. 1, 2, 3, 4, 5 in the ribose, and 1, 2, 3, 4, 5 in the deoxyribose. And if you were to compare the structures, you'd see that the only difference is going to be at the 2' location. And it's this carbon right here. So whenever we're talking about 2', we're talking about the second carbon in the sugar. So right here in the deoxyribose, there's just an H, and in the ribose, there's an OH. And that's the difference between those two. And the way that I like to remember it is just kind of looking at the word deoxyribose. You'll notice that ribose is the same as this ribose. And the only extra thing that's added to the word is deoxy, and oxy is like oxygen, so deoxygenated. And if we were to compare OH and H, it's like you just took an oxygen away. So we deoxygenated the ribose to get deoxyribose without that oxygen at the 2' location. But you'll notice at the 3' location, both have an OH. And that's in a really important spot. So the difference is at the 2' location. Ribose has an OH, deoxyribose has an H. All right. So now we're going to go ahead and start talking about the bases. And before we talk about the differences with DNA and RNA here, I'm first just going to talk about what they look like. So the bases can have 2 rings or 1 ring. If they have 2 rings, they're called purines. So they look like this. So you'll see here's the first ring, here's the second ring. And if they have 1 ring, they're called pyrimidines. So here's just 1 ring in the pyrimidine. And the way that I remember this is that the longer word goes with the shorter structure. So the longer word, pyrimidine, goes with the shorter structure. The shorter word, purine, goes with the longer structure. All right. So now let's talk about the different types of bases that you might see. So in purines and pyrimidines, there's going to be a couple of names that fall into each of those. For purines, the 2 bases that you need to know are adenine and guanine. And the way that I remember this is angels and gods are pure. So A for angels or adenine, G for gods and guanine are pure. And that means that adenine and guanine are purines. The next one is pyrimidines, and the 3 here are going to be thymine, cytosine, and uracil. And the way that I remember this is TCU is not pure. So we have thymine, cytosine, uracil, and if it's not pure, that would mean that it's not a purine, it must be the other one, they're pyrimidines. So now let's kind of start looking at the differences between DNA and RNA. In DNA, you have adenine, guanine, cytosine, and thymine. While in RNA, you have adenine, guanine, cytosine, and uracil. So the difference in the sugar between DNA and RNA is that RNA has an OH at the 2' location and DNA has an H. The difference in the nitrogenous bases between DNA and RNA is that DNA has thymine and RNA has uracil. And the other 3 bases are the same in both. All right. So now let's talk a little bit about DNA versus RNA strands. So generally, DNA is going to be double-stranded and RNA is going to be single-stranded. So for your DNA, that would mean that we have 2 strands, and that means that you're going to have some stuff in the middle interacting. The stuff in the middle interacting is always going to be your nitrogenous bases. And that means that some nitrogenous bases are going to always be pairing together. So let's talk about those pairs. A is always going to pair with T, so adenine is always going to pair with thymine, and G is always going to pair with C. Guanine always pairs with cytosine. And the way that I remember this is apples grow on trees, gas goes in cars. Apples grow on trees, A and T go together, gas goes in cars, G and C go together. And it's also helpful to know that a purine and a pyrimidine are always paired. So remember, a purine, that's going to be your adenine and your guanine. And your pyrimidines, that's TCU. Since we're talking about DNA, that means there's thymine instead of uracil. So I'm not going to talk about uracil. So you'll notice that for the pairs, it's always going to be a purine with a pyrimidine. So you have the adenine with the thymine, the guanine with the cytosine. All right. So there's a couple of bonds that you need to know for DNA and RNA. So the one that's going to be in both DNA and RNA is your phosphodiester bond. And this is connecting your sugar and your phosphate. So it's going to be in your backbone. And we'll look at the backbone in a second. The second bond is really important with DNA. It happens in RNA too, though. So in DNA, we talked about how we have that double strand and you have your nitrogenous bases in the middle. And the way that they interact with each other is through hydrogen bonds. So you would have an adenine and a thymine that would interact with 2 hydrogen bonds, and a guanine and a cytosine that interact with 3. So the way that I learned this was A2T G3C, A2T G3C. And that is what is always playing in my head whenever I'm thinking about the hydrogen bonds. If that doesn't work for you, you can just memorize it. The reason it works for me is that like G, 3, and C, they all end in that E sound. And I know that A and T are paired together, and T and 2 start with the same letter. And for me, it's just kind of like a rhyme in my head, A2T, G3C. So if that works for you, that's great. But also it's not crazy hard to memorize those numbers. So if that doesn't work for you, feel free to just memorize. There's 2 hydrogen bonds between adenine and thymine, and 3 between guanine and cytosine. All right. So the last thing we're going to look at is just a little picture of DNA and a couple things about it. This is going to be important in later chapters because chapter 10 really is a building block chapter. So first, let's talk really quick about how they're complementary. They're complementary because you always have your T with your A, your G with your C, and so on. And now we're also going to talk about how they're anti-parallel. So there's 2 ways to think about it being anti-parallel. Go with whichever fits with your brain best. So if we were to start at the top for both of these, the one on the left goes 5' to 3'. The one on the right goes 3' to 5'. So they're opposite. The other way you can think about it, if this works better for you, is that traveling from 5' to 3', one of them goes down while the other one goes up, 5' to 3'. So whichever way works better for you to remember that they're anti-parallel, that's great. Now let's talk about what that 3' and 5' mean. So the 5' end is going to be your free phosphate end. So there's just a phosphate right there, and you're not able to grow off that end anymore. You can't add any more nitrogenous bases. And that happens right here and right here. And your 3', this is where the next nucleotide is going to be added. So remember, a nucleotide consists of that phosphate, a sugar, and a nitrogenous base. So one would be added right here, and one could be added right there. And that's where it would grow. So DNA would always grow in the DNA always grows from the 5' to the 3' direction. And that's because you can't add anything onto the 5' end, and you always add on to the 3' end. So everything goes from 5' to 3' one way, 5' to 3' the other way. And that is the end of this tricky topics video. Really make sure that you have this information all down because it does become very important in the later chapters.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.